I'd like to now introduce our final speaker for this session. This is Dr. Michael Milham. Michael is an internationally known neuroscience researcher who is currently Vice President of Research and Founding Director of the Center for the Developing Brain at the Child Mind Institute. He has a PhD from the University of Illinois at Urbana-Champaign in cognitive neuroscience, and has produced more than 140 publications related to fMRI. And these have come from research projects examining structural and functional connectivity in the brains of developing, aging, and clinical populations. He is interested in advancing a big data research agenda in neuroimaging, and that focuses on open data sharing. So today he is here to talk to us about the PRIMATE Data Exchange, which is an open science resource for non-human primate imaging. And welcome, Michael. The session is yours.
Okay, thank you for inviting me here, and I appreciate the opportunity to talk even virtually as the situation has it. I want to give us a little bit of perspective as to why you have someone who's a child and adolescent psychiatrist talking to you about open data sharing for non-human primate imaging. I think over the course of my talk will become more apparent, but just in the big picture, my background, as was just said, is I'm a neuroimaging researcher, also a child and adolescent psychiatrist. And really one of my focuses over the years has been, how do we sit there and make the research efforts going on useful? How do we make them most valid, most reproducible, and really something that will one day yield clinical deliverables? In my case, for interest in psychiatry, but of course there's a broad range of applications. And coming as someone from the functional MRI community, it's a community that's gone through that evolution of having very small studies initially on the human front, with then increasing the scale and then realizing the need for open sharing and open science in order to advance an agenda that pushes us towards a more reproducible science with the potential to one day have clinical deliverables. In the course of my journeys, I also picked up an increasing focus on non-human primate research. When we look at a lot of the questions that we'd like to answer about novel interventions and therapeutics, animal models are essential. And from that perspective, that got me to start looking at this community and becoming involved in this community. Now, as someone coming from a human community, we've gone through the stage of being a cottage industry and have been growing over the last several decades. And really one of the things that came through is the many unique challenges beyond everything that the human imaging community has faced. The non-human primate community faces even more immense challenges associated with the many of the nuances in non-human primate handling, in many of the different intricacies of how the small brain interacts with the scanner, and really just the reality that that the many of the tools being used were initially designed and optimized for human application. And so that really has become a major focus of mine over the last few years, is how do we bring that same process that's happened in the human imaging community, which is still evolving, but how do we bring that more towards the non-human primate community? And so you'll see with what I'm going to go through today, I would first introduce the topic of open science in non-human primate imaging. Then what I will do is look a little bit towards what's been accomplished in the human community and kind of the agenda that open science brings about, and then introduce some recent efforts in the non-human primate imaging community that I would argue are pushing towards greater implementation. And I would have to thank both of the prior speakers for doing a fantastic job and making my life easier, given the great job they did in introducing a lot of these concepts. Okay, so for starters, why open sharing, data sharing is needed for non-human primate imaging? Well, as I said, non-human primates remain a crucial animal model going between rodents and humans. And I think that most would agree that we do need some intermediary model between the two extremes, as far if we really want to maximize our understanding of human brain function and be able to translate some of the systems neuroscience findings we get from the rodents towards the human. The NHP community, it has a broad range of species, and each, unlike human studies, there's incredible investment in each single subject in animal non-human research. So there's an incredible amount of investment. There's a wealth of diversity, and it's a challenge to not, if we don't work to pull it together. Collaboration coordination is, and once again, if we want to maximally use the data, if we want to make the most of what's available, and shift from a cottage industry style environment where folks are having studies with maybe a couple non-human primates or 5 to 10 to suddenly start talking about numbers that are much more appreciable, collaboration coordination is essential. If you look about 10, 15 years ago, the human imaging community would be considering 20, 30, 40 subjects a substantially sized study. Now people want to hear about hundreds and thousands. That is a culture shift. And that culture shift is made heavily possible by collaboration and coordination. No single group is going to do that. And even if a single group could do that, it would engender an environment where we would then have an incredible amount of unnecessary redundancy. So when we look at non-human primate imaging, where researchers are each working very hard to deal with the intricacies of their subjects, it's that process of pulling it together that is essential to really make the most of the data that's being collected. Open sharing fosters transparency and reproducibility. This has been well established. I think the prior speakers have addressed that point heavily. And one of the things is we need a greater coordination of work together, even to support the issue. We need to work together for how do we deal with issues like crediting groups who collect the data as opposed to those who are analyzing the data, or dealing with some of the licensing questions that come up with data sharing. And then finally, by opening up non-human primate imaging to a broader audience, what you do is you invite human imagers into the picture. There are lots of very talented imagers and analysts who will never be able to collect non-human primate data. And in a closed community, they will never be able to contribute their many analytic skills and abilities or insights and knowledge to the equation. Once you open up the data and make it openly available, what happens now is you invite a much broader workforce in the scientific community. You create these links between different parts of science, essentially. It becomes an exciting landscape. So let's just take a quick look at some of the lessons we learned from human imaging and then showing how we're trying to translate them into non-human primate imaging. So in human imaging front, back in 2009, we founded what was called the 1,000 Functional Connectomes Project, which was really about 1,300 data sets shared across about 30 sites. These were data sets that were previously acquired independent of one another. Everyone agreed to just contribute their data. We organized them, arranged them in a reasonably fashion to be shared, and then using a NITRIC, which is a NIH-funded resource, we made it openly available. So suddenly researchers who were struggling to get 20, 30 data sets to test out their analytic techniques and so forth had over 1,000 data sets. They also had a way to start trying to figure out what's reproducible in findings across different sites and what is not, which is an incredibly important question. Since that time, we founded the International Neuroimaging Data Sharing Initiative, or INDI, which has had initiatives like the ADHD 200, which suddenly armed folks who've never touched imaging data with data sets that now contained almost 1,000 children, about a third with ADHD and the remainder without, so they could start asking more valuable neuroscientific questions, as well as coming up with diagnostic methods and so forth. There's the Autism Brain Imaging Data Exchange, which has shared over 2,200 data sets across probably about 30 sites at this point, half with autism and half without. And then CORE, the Consortium for Reliability and Reproducibility, where we shared a whole range of test-retest designs. So to push the community towards thinking about the issue of test-retest reliability in their measures. That was almost 5,000 data sets from about 1,500 individuals across probably, I think it was about 15 sites in that one. The point is, these are examples of initiatives where these are all grassroots initiatives where people have independently collected data, but found a value of sharing the data as a collective and being able to make that openly available to the community. So one of the course questions that comes up, and this is where I think the prior speaker kept referring to carrots, is, what's the impact of this? Do you have an impact on the literature? Can we show that shared imaging data really impacts life? So here's a paper that we published in Nature Comms, where We first of all show across 5 different initiatives the uptake of the data in terms of publications generated by independent groups with the openly shared data. We also note, even though a lot of folks will worry about, oh, what about the younger investigators? There are actually 58 theses at that time. This is back in 2017 with that were written using the openly shared data. There's also, if you look as far as the journals, this is all bibliometric analysis. If you look at the journals in which the data is being published, you find a very broad range of classes, from general neuroscience to neurology, general medicine, computer science, mathematics, imaging technology, engineering. The vast majority of data was collected by psychiatrists, psychologists, and a small number of neurologists. So it's showing that by making the data open, you recruit a much broader audience and the analytic capabilities of that audience and the collective workforce. There were over 1,000 publications as of 2017 with continued growth. And with some of the initiatives like for Autism Brain Imaging Data Exchange, there were, as of about a year and a half ago, there were 2 papers a month being published from that data. That is an incredible rate when you think about what would happen otherwise. What would happen otherwise is those data sets would have sat in forms of 30 to 50, maybe 100 data sets in each individual lab, each underpowered for what they were after, and each publishing at a much slower rate with more limited focus. This here is a plot just showing the co-authorship patterns that were observed in the bibliometric analysis. It really emphasizes the global nature that's achieved through open data sharing. And this is an important slide, and that what we're showing here is for any one of these initiatives, if one were to price out, what would it cost for me to have come up with the equivalent number of publications had I used all newly collected data, as opposed to having used openly shared? The conservative estimate was about 800 million. The less conservative estimate was about $1.5 billion is what it would have cost to have every one of those studies using newly collected data. So there's an incredible return on investment here, even when you go through and try to deprecate the value of the data and so forth. And one of the things is, depending on the data set, for a more minimal data set, it might cost $1,000 a participant. But for some of the more complicated data sets, like the ones where you're looking at autism, children with autism, it could be up to $10,000 per data set. So the reuse of data is incredibly valuable in this circumstance. Looking at the non-human front, the costs are even greater if we think about how much goes into the maintenance and support of data generation of a subject's worth of data. Another thing that emerges in this environment is an increasing focus on best practice and standards. And this is something I think the prior speakers really did a nice job of driving home, is the importance of focusing on standards and really trying to improve the rigor of the community. And in the imaging community, there are just so many advances that have been going on in the open science front, but a particular focus has been on even just going back and taking a step back and thinking through how we arrange our metadata. The BIDS format has emerged as a dominant one in the community now for organizing the metadata in a more meaningful standard that could be readily shared across tools. And one of the mistakes that folks have been trying to correct compared to earlier years is there have been prior standards used and still are used in the imaging community. But one of the challenges is having a standard is not enough. You have to have agreement on how to implement the standard, because otherwise you create a standard that different tool makers implement somewhat differently, and you watch these difficulties emerge in the community. And we've seen this. So that's something that now, by having, for example, in BIDS case, there's a BIDS validator, so is there compliance with implementation of the standard in a given data set? These are significant steps forward. And also there's been a lot more forced thought over how do we go about crediting folks in an open community. So in the paper that listed on the lower right is one we wrote some years ago, really thinking through data descriptors and pushing the agenda of data descriptors in the imaging community. This idea that if you share data, you should be able to get credit for sharing that. And data descriptors are one of the publication-based ways of crediting folks. Another thing that emerges from all these efforts is the opportunity to think through data cleaning and harmonization. When you have data sets from a range of scanners and a range of of different acquisition strategies, you could start looking for trying to deal with batch effects. You could start looking for common nuisance signals. Of course, with fMRI, our biggest enemy in the world is typically head motion, probably followed by respiration and physiologic signals of that sort. Well, but one can really start to work through these issues and think about how these phenomena are showing up in different protocols, and really what are those approaches that could be used to remove batch effects. And I'm saying you wouldn't get this if you didn't just openly share the data from a range of sites, because otherwise this would be a far slower process, and one would have to think to even go after this and then find the sites to do in a coordinated study and so forth. And the speed of science and advancement would have been far slower than what it's been in the last decade of the neuroimaging community. Another thing that emerges is increasing focus on reproducible analysis pipelines. And here's just a small number of examples of openly shared pipelines that are trying to increase their focus on and scalability, on increasing data quality, also on kind of pipelines that are more robust. And there's a whole story that's emerged over the last decade in the neuroimaging community, where on the one hand, and I know there's just a recent paper discussing from Russ Poldrak in Nature discussing the many different decisions that neuroimagers will make to focus on it when focusing on a single data set. On the other hand, as these tools emerge, we can either have folks use a more prescribed turnkey pipeline or one-click pipeline, I should say, similar to fMRI prep. Or what one can do is when you look at we have a pipeline that's CPAC, or the configurable pipeline for analysis of connectomes, where it allows you to make a whole diversity of decisions, but everything is tracked in a way that your analysis can be easily reproduced by another lab just by using a config file. So there's an increasing focus in CPAC as well as these other pipelines in moving towards inclusion of optimizations for non-human primate imaging and rodent imaging. And this idea of reproducible analysis pipelines is essential, and it's one of the key ingredients for moving from a cottage industry to a reproducible science. Okay, so now switching to focus to the non-human primate imaging front. Back in 2018, we launched what was referred to as the PRIMATE Data Exchange, or PRIME-DE, which we put a data descriptor for in Neuron, and showing the article here. This resource pulled together, and here's the webpage that one can go to on the INDIE website and go and just actually immediately start downloading data from the PRIMATE Data Exchange. There are initially there were 25 samples from 22 sites, which I would have to also point out, actually there was almost half that when we started writing the manuscript. But as more and more word got out, more and more individuals started offering data and contributing their data. So what happens, we went up with over 200 data from over 200 subjects that were, a combination of structural imaging as well as resting state fMRI for a large number of them, some with naturalistic viewing or task fMRI, as well as diffusion imaging. So suddenly someone who has never touched non-human primate imaging data before and would never have the opportunity to collect such data now can go online and download this data and start working with it. And then also labs that are contributing, they might be contributing, if you look at the numbers, 4 to 10, for some of the larger samples were 22 or 19 subjects. Suddenly they now have access to data from a couple hundred subjects. So if you've contributed, what you're getting back is more. If you're on the outside, what you can offer is your skill sets and knowledge from other disciplines. It creates an environment that we take away a lot of the waste that happens in science, and we start to deal with this reproducibility challenge by actually equipping scientists with the data they need to ask questions about reproducibility. And one of the things that immediately one can do is, here's examples where one can now suddenly start looking at images from different scanners. I will tell you the amount of work it took to get this, a pipeline together to handle the many different nuances of different scanners to create this figure was not trivial. But the point is, once you have that from all these different scanners, you can do that work. Even right now, there's one of our investigators who has been working on deep learning algorithm and in their application in this data set to be able to have automated brain extraction, which brain extraction is the single most challenging step, and also tissue segmentation for most pipelines in imaging, going back to human literature which that problem has been largely addressed, to the non-human literature where folks wind up having to do lots of manual tracing. Everyone is tweaking human-based tools for the specifics of their data set and the variations in data quality. Now suddenly you have an environment where one can use deep learning algorithms and other approaches and train on a whole range of data sets from different scanners and come up with more generalizable methods that would dramatically reduce the work effort and also improve the quality of the research that's yielded. Here's examples of functional imaging. Another thing that could be done is one could take automated QC metrics that have been developed in the human imaging community and start to apply them to non-human, look at variations in data quality, varying metrics across sites, figure out which ones matter, which ones don't, start to think through how to change these metrics in order to better address the challenges of non-human primate imaging. And here's a first report that come out from our team where we were able to take the few larger data sets that were in existence and actually go and map out inter-individual variation in the brain in a manner that had been done just about 5 years prior in the human literature, and look for similarities. And we were able to look for what are the similarities across data sets in the non-human, as well as, of course, non-human to human. And the other thing we were able to ask is, what are the differences between awake and anesthetized states? Most of the functional imaging to date has been done anesthetized. There's an increasing focus on awake. And one of the things that we've done in a few different articles is showing how profound a difference, both awake and anesthetized, both are highly reliable states when you have sufficient amounts of data, but they're different. They're not equivalent. And which that shouldn't be surprising, but often in the literature, people use these things interchangeably. And this allows us to start asking those style questions. One final output that grew from the initial effort was the idea that we should have a meeting, that we should actually get folks together beyond a set of conference calls. And so what happened in actually just this past fall was the PRIMATE Data Exchange's Global Collaboration Workshop, which pulled together about 90 investigators from around the world at Wellcome Trust, headquarters in London. And really, what we had was 2 days of folks just, rather than having any sort of formal talks, it was 4 different breakout sections, and in which every person participated in every breakout session. So they were done serially. And we were able to address or identify what is the agenda, what are the challenges. And what this yielded was a few things. First of all, there's a paper in Neuron that you're seeing here that really summarizes all the many different discussions. We looked at everything from standardizing data collection to animal welfare regulations, intellectual property, to data standards, quality assessment analytics software, and coordinated paradigm design. And the thing is, folks were able to realize what are the things that are in common and what are the things that are different. So, for example, in common, when looking at the data collection, there was general agreement pretty quickly on minimal data acquisition specifications. So minimal voxel sizes to use for structural imaging, minimal voxel sizes to target for functional imaging, and also then talking through how do we address those gaps that between the ideal and where each of us felt we're at. With anesthetized imaging, we were able to quickly find out what are the common practices that emerged for anesthesia protocols among the studies, and then identify the differences. And awake imaging, folks were able to really scope out what are the challenges that we're having and start to share ideas about behavioral training and then how do you deal with some of the mechanics of awake imaging. Animal welfare regulation and intellectual property, that was a very robust set of discussions. Really just recognizing the challenges that differences in regulations across different nations have, how that impacts open data sharing into scientists, and also discussing some of the challenges in how folks approach engaging the public, where several admitted to being their institutions were more prone towards hiding, and and then those who've been through a variety of experiences were explaining the value of openness and promoting transparency, and identifying the UK Concordat as an excellent example of getting non-human work into a space where there's open public engagement and transparency to dispel many of the myths or concerns that exist, and also have an honest engagement with the public. And then issues regarding crediting intellectual property, how do you deal with priority on publishing for shared data, and protecting interests while at the same time keeping things open as possible and promoting collaboration. And you know, this is something that if you look through that meeting report, it's a relatively quick read, but it really contains a wealth of discussions. And of course, then, like I said, I mentioned data standards. Some of the discussions, some of the earlier talks mentioned the many forms of metadata that need to be accounted for. And when one looks at it, it's doable in non-human primate imaging. But what we'd have to do is take some of the constructs created for human and modify them. And it's possible. It just needs to be created as a common agenda. And that's very much where this meeting took the attendance. One of our outputs that immediately followed this meeting was what we called a brain hack. This is something that's been in the human community dating back to about 2012, where it's a mix of an unconference format and a hackathon. And it's intended to bring together a range of folks from neuroscience, psychology, to your computer science and so forth. So it was a 2-day event where folks work together. They just identify spontaneously projects to jump into. They start to team up, show each other what they're working on. You'll notice the one circled on the bottom of the screen is Brain Hack Comparative MRI. That's what followed the Global Collaboration Workshop. And when you put folks together for 2 days, you now help give really a chance to be interactive with one another at another level beyond what you got from the workshop. And just as an example of an output, here is the PRIMATE. So now there's a resource exchange being built. So we have the data sharing exchange, and then here is the resource exchange that's really meant to being a share of knowledge and to share scripts and analysis resources. So this is something that's been being constructed. And there's actually also a special issue in their image that'll come out within the next year or so, where the many attendants from the consortium and workshop have been working together to write a series of collaborative articles. So again, fostering collaboration, communication, exchange of ideas, bringing about transparency, and really trying to accelerate the evolution of non-human primate imaging from a cottage industry with a large amount of redundancy and samples that are too small to feel confident about reproducibility towards a reproducible science that maximally uses the data, maximally ensures its impact by inviting a broader community, and really works towards just optimizing science. And I just like to say thank you to the PRIME-DE organizers. In addition to myself, there's Daniel Margulies, Chris Petkoff, and Charlie Schroeder, and of course the many different participants from PRIME-DE. And that's it.
Thank you so much, Dr. Milham. Fascinating that there's so much data sharing happening now, and I do think that there's so many opportunities for us to continue learning and also apply, the 3 R's that we've been talking about for the last couple of days because of the wealth of data that's available.